we decided yes to make this webinar in English. So just a few words like to introduce what is GCC. So yes, this webinar is organized in the framework of the Global Cosmetic Cluster. So it's a global network representing the entire value chain of the beauty industry. So like we are dealing with ingredients, formulation, manufacturing, packaging, finished product, distribution and logistics, and other topics as well. But it was to tell you some of the main aspects like the network is dealing with. So this network has, let's say, four main dimensions and four main goals, which are like the first one is like to support the internationalization of the SMEs. So I will switch to the next slide. Yeah. So to support the internationalization of SMEs in the cosmetics, aromas, and perfume sectors. So many dimensions are like we are working on many dimensions, such as export or, for instance, finding new partners. The second point of the global cosmetic cluster is to ease the access of cluster members to international experts in the field of cosmetics, aromas, and perfumes. So clearly, like it's the access to some experts is easier through this network. So don't hesitate to ask the network for finding such experts. The third point is to find your business partners or innovation partners among all the cluster network. So it can be, for instance, like missing skills or technologies. And the form is available on the website to find your right partner. So don't hesitate just to check, online. We will send you the slides, and I will put, the link of the website. And on this website, you have a form which you can fulfill and find the relevant partners, like for your business or for your innovative projects. And the last point is like we collaborate between clusters in the cosmetics, aromas, and perfume sectors for the benefit of all our members. For instance, it can deal with like getting funding from European Union and distributing these funds through vouchers. So it can help, through funding like an innovative project of our members. And then also it can be like, as we are doing today, to organize webinars on specific thematics. So these webinars are also like an occasion like for the network to be aware like of the last development in the cosmetic industry. And it's also a place, like for animating a community. So I will switch to the last slides for myself. And here you can see the next dates with the global cosmetic industry webinars. So today deals with like smell, odors, and COVID-19. And you can see like in coming next months, we will have something about natural ingredients and related regulation organized by Bioentropic, the cluster Bioentropic. We will have also, like the new materials organized by the cluster from Taiwan. And then in September and November, you will have something, a topic dealing with the supply chain and then with the packaging industry. So if you have more questions about the GCC, the Global Cosmetic Cluster, I will put my contact details in the presentation you will receive. And you don't hesitate to contact me if you have any other questions regarding this this network. So now I will give the floor, to Philippe Clos. He will present Innov'Alliance, and he will do also a quick overview of the fragrance and flavors market. So thank you everyone, and have a nice webinar.
So thank you very much, Clément, for this kind introduction. And welcome to members and all partners to attend to this webinar. In the recent years, the smell and senses were highlighted by the pandemic crisis, and notably the possible alteration of the senses. And that is, and as we know, it is very important for professionals of the industry to have a good functional taste and smell senses. We decided to treat this topic in this webinar, and for that, we are lucky to welcome some big experts on the senses with professor of University Manchester, Matthew Cobb, Miss Charlotte Sinding of INRAE in Dijon in France for taste sense, and Valentina Parma from the the Monell Institute, which present us some results about alteration due to COVID-19 infection. As told by Clément, I am going to present you briefly the competitive cluster Innov'Alliance, and then a brief overview market of fragrance and flavor market performed by our market survey service. So Innov'Alliance is almost a newborn competitive cluster since it results from the merging between Terralia cluster, which was mainly focused on agriculture and food issue from the main cultivation in southwest of France, and PAS cluster, which was based in Grasse and mainly concentrated around flavor and fragrance industry and cosmetics. So nowadays, Innov'Alliance is therefore a unique network and a big competitive cluster, which groups more than 430 members. Among them, we can find more than 300 companies. The new cluster Innov'Alliance has a big ambition, which consists to support ecological transition of agriculture toward 4 main application sectors, which are food, nutraceutical and health ingredients, cosmetic, flavor, and fragrances. To overcome this challenge, we have 3 main missions: a core skill, which is focused on innovation to stimulate and support innovation, but not only innovation, it consists also to support new products and services on the market with some business development mission. And all of these missions were performed in the order to improve environmental and social performance. Briefly, if you look at the organization of the cluster, it's a classical vertical organization according to the value chain, from the raw material and notably the agricultural side, but not only, because green chemistry or biotechnology ingredients are welcome among the production of the members of our cluster. And we can see on this slide some members of our cluster as well. It can be some manufacturer of ingredients and notably natural ingredients, like the leading company Robertet, for example, or Naturex Givaudan. And also we can find also some company which formulate and assume the distribution of new products like Arco Pharma or L'Occitane en Provence or the other on this slide. And complementary to this organization, we have a strong pool of suppliers of technology, which can be expert, laboratory, equipment supplier, or specific developer of new captor or something like that. And if you join the cluster, we can benefit from a personalized support, which can help you to use a complete pack of services around all 3 missions. So you have main mission services on this slide, and we send you the presentation. Nowadays, I propose you to present a market overview of the flavor and fragrance industry, an overview performed by survey and intelligence economic service. And first of all, if we look at the organization, as everybody knows, fragrance and flavor is at the heart of an important value chain, which generates high value as well from an economic point of view, with important turnover and people workers, but also in terms of social value. The last year was a good year after the difficult 2020 year, since, as we can see globally, the aromatic production is in progress with a growth of around 8%. Recently, the French trade union Prodarome has published the results from the French industry, which shows a growth of more than 10% according to the different segments of production. And concerning the end market, as we can see, it is only the main end market because there are many applications, and food is a too wide market to precisely define one rather than another. All these end markets are in growth, show strong growth between 4 to 8%. So it means that we have almost recovered the level before the pandemic crisis, or sometimes take over the previous turnover. If we look at more precisely different markets, concerning the fragrances market, we can distinguish two major trends. And it was also the case for all the markets. There is a real impact of the crisis. Impact on consumer behavior, and notably of the acceleration of some trends like eco trends. And it is clearly the case for the fragrances market, with the naturalness shows strong growth, and simultaneously brands are innovating in many different ways to help drive engagement with fragrances, from tapping into wellness trends, helping people create bespoke scents, and promoting eco-natural and free forms from merits. Concerning supplying of fragrance, it means that for ingredients, the top 5 fastest growing claims are natural, ethical, and eco, and you can see after upcycling and alcohol-free formula are also in strong growth, and botanical ingredients clearly dominates the new formula. Concerning the creation, playful packs, disruptive collaboration, and customized and versatile formula help fragrance brands stand out in store and online. So it means that the perfumers and perfumes should help customization and produce emotions, bring comfort after the difficult period that everybody has known. And for example, some illustration of these trends, if we look at the main claims concerning fragrances ingredients, we can see that botanical, that almost natural and eco have almost doubled. It was the case for botanical ingredients. Ethical, sustainable, packaging is a clear point, and vegan is in strong growth. And you have also in this site some example of how brands use and tap into these new opportunities. For example, with this with a Bulgari product which creates a joyful scent experience based around emotions. This package, for example, comprises of 5 eau de parfum in colorful bottle and 5 magnifying essences in all white flacons, and everybody can combine with the parfum or wear alone. You can also find some brush on tube, which causes pleasurable feeling on application. Concerning the hair care market, consumers are increasingly looking for more sustainable and eco-friendly hair care products, and seeking out hair care which provides them with aromatherapeutic benefits. Meanwhile, brands are tapping into consumer demand for odor and bacteria-fighting hair care also. And brands are also turning to natural ingredients to mitigate consumer concern over the chemicals. And concerning issue from these major trends, the opportunities for fragrance suppliers are still to produce ethical, environmental, natural and functional ingredients. It is notably the case for odor neutralizing and antibacterial ingredients. Botanicals are always the main ingredient, as we can see just after. Concerning the creation, the perfumer have to engage consumer in fun, exotic, and destination fragrances to help the home atmosphere improvement, notably in period of lockdown, for example. We can also measure a decline of gourmet edible fragrances, and some illustration about these tendencies. For example, in Asia here, you can see that odor neutralizing claims and antibacterials are a significant part of launching, since it is around 20-25% of launches for odor neutralizing agents, and almost double on the last period for antibacterial activity. You have some products here to illustrate these trends. For example, the combination with natural extract, and notably for their bacteria fighting activity associated to free deodorizing agents for a strong deodorizing effect. You can also find new fragrance profile labels, and here you can find some labels associated to some colors like mood, like fresh and relaxing, floral uplifting, and fruity happy. The household products, consumers are increasingly expecting brands to paint a more holistic picture of their eco-credential, from convenient refill options to carbon neutral production and use of recycled materials. Meanwhile, antibacterial properties remain a priority for consumers. As a functional category, consumers expect household cleaners to not only clean effectively and remove unwanted odors, but also to provide pleasing and long-lasting scents. So the challenge for fragrance suppliers are to produce, of course, sustainable ingredients with high priority for disinfection properties, and ingredient safety also is a big concern. In terms of creation, new and interesting fragrances to engage consumer in this anxious period, and where time press period also, where the clean product help to save time. And if we want to have some illustration about that, concerning emotion and freshness, we can see that citrus and fresh and clean scent show an uptick in launch activities, around 10% for these main notes. And you have some example of products, for example, the use of natural and recognizable ingredients to reassure consumer of safety. For example, that one with 97% of natural ingredients. So it is the picture, it's natural, so it's safe, even if everybody knows the limits of such thing, such thought, for example. And we have also some great composition house, which produce new fragrances for household consumer engage, ici Maison de Parfum, in collaboration with Odense. So briefly, if we want to resume the major trend on the fragrances, or on the main fragrances markets, we can clearly observe an impact of the pandemic crisis, which causes some acceleration of some trends like eco, natural, sustainable trends, but also the need for consumer to find many feelings of comfort and reassurance through smell. And these trends observed for the fragrance markets are of course also observed for the food and drink market. Concerning food and drink market, the uncertainty and precariousness issue from the pandemic enhanced the need of a sense in control for consumers. They need to be in control to make confident decisions that protect the health and the health of the planet. consumer will also be seeking joyful products that amplify the flavors, colors, texture, aromas, and interactivity that food and drink can provide. And brands can, for example, provide opportunity for playfulness at home, and we can find meal kits, digital cooking classes, or gamified recipes to give more fun during this period of social restrictions. And then retailers, restaurants, and brands can create multifunctional and meaningful spaces where consumers can connect, shop, and eat, both in person and online. These major trends imply some opportunities for not fragrance but flavor suppliers. We can see the emergence of new flavors in beverage concentrates, for example, and concerning creation, ethnic flavors and authentic street drink. The absence or reduction of social life during the pandemic crisis has led consumers to bring many of the pleasure associated with out-home consumptions into their homes, and this context has caused an increase in cocktail making, also known in mixology. Thus, beverage concentrates explore cocktail-inspired flavors. Berry and citrus, for example, remain the most prominent flavors in beverage concentrates. And if we want to show some illustrations of these major trends in Brazil, for example, as everybody knows, flavor is a crucial purchase driver for yogurt consumers in Brazil, and as many, 56% of categories users cited indulgent flavor as an important attribute to buy a yogurt. And you have on this slide the main flavors bought in Brazil during this period. We can also measure an increase in the meat and meat flavor snacks. So it is the need to create at home some playfulness. And previously I talked about mixology, and here you have an example of innovation targets in-home mixologists with Irish cream, vanilla, amaretto, and so on. So, and a last point, a more quicker overview, and we can measure a decline in gourmet notes in healthcare, but concerning fragrance, we see more and more food and drink inspiration in fragrance and scents. For example, in China, a creation of 3 flavors from drink collection, including matcha green tea and cheese milk, for example. We can also see concerning hair care, McDonald's, McCafe launched 5 scented candles smelling like it's coffee, a fair representation of the countries where the coffee beans were sourced. And last innovations, the Idaho Potato Commission created a perfume made from distilled Idaho potatoes and essential oils that are intended to smell like chips or fruits in French. So it could be strange. We can see also appear some champagne note or something like that. And there is a beginning of growth concerning this crossover between drink and fragrance. So here it is for this quick overview about the flavor and fragrance market. If you had, I don't precise at the beginning of the webinar, if you have some question, you can use the chat, and then we publish your question. And I propose you to put your question on the chat, and we publish all the questions at the end of each presentation. So, are there some questions? Okay. If the presentation will send you after the meeting, and if there are not presentation, we'll begin by a presentation about the smell sense by a big expert, and Mr. Matthew Cobb present us how the nose knows in you a maggot and a Neanderthal.
Bonjour tout le monde. J'espère que vous m'entendez bien. J'ai vu dans le chat que certains ont eu des problèmes. I'm going to speak in English, but as I can speak French. The maggot, by the way, is l'asticot, and so you'll see that we're going to get quite an interesting range of animals that can smell. So I'm a professor of zoology, so I'm interested not just in human smell, but in the smell in all animals. And what I'm going to do is to provide the background for Charlotte and Valentina's talks, which are going to go into much more detail about smell and in flavour, how they're mixed, and also the effects of COVID-19. So this is going to be just some basic science, giving you some examples from my work. So the first question as a scientist you want to ask is, well, why do animals smell? And of course, it's not just animals. Bacteria have got a sense of smell, but let's just focus on animals. They want to find food is fairly kind of obvious. They also use smell for memory. So in all animals, when you have a smell and you detect a smell, two things happen to it. Firstly, it's put into part of your brain which is identifying exactly what you have smelled. But the second part, it's put together with other events that are happening around you. So it goes into memory. And this picture here is an example of a memory for me that can be evoked by the smell of this smell or taste of this drink, which is a very sugary drink that we have in the north of England, or these sheeps, these crisps from the UK, which had a cheese and onion flavour. And if I smell or taste either of those things, I am immediately taken back to the world of the swimming baths, la piscine, where I would go as a child and then afterwards have these this hot drink and these crisps. So memory, we know this from Proust and so on, that you can reactivate memories in a very powerful way by the use of odor. And that is because of this separation of the signal right at the beginning of the process, one into identification, the second into whatever is happening around you. And then the memory can be formed with a tag of a smell or a taste, and that can be then reactivated. You can also, of course, use smells to find a mate, and this is very common in the animal kingdom. Here we've got two beautiful flies. Sad to say, we don't get these in Europe. These are from North America, and you can straight away tell which is the male. The male is the one with these astonishing antennae. This is the female. She's got much smaller antennae. And so the male has detected the female's pheromones, these chemical signals which are given off, and has been able to find her and mate. Now, there are no known pheromones of any kind in humans, although there are certain substances that are sold as sex pheromones, and I'll be coming on to them later on. Why there might be no pheromones in humans is a big matter of debate at the moment. It's possible that there are things similar to in rabbits. There's something called the nipple search pheromone, whereby the baby rabbit has to latch onto the mother's nipple very quickly. It's possible similar things exist in humans, but they have yet to be demonstrated. And of course, you might want to identify your home with your smell. This may be very important for finding your way home, as in the case of bees. Or if you're a dog, you might want to identify your territory by producing a whole set of signals that are associated with you that aren't straightforwardly a pheromone, but are more some kind of signature. And very strikingly, although animal marking is incredibly common, we know very little about the kinds of chemicals that are associated with it. And you might also, of course, want to be able to detect danger. One of the most significant smells throughout the animal kingdom, which produces a very strong response, is the smell of smoke. And this is why if you have a fire in your house, you will wake up, because that smell is going to activate very deep circuits inside your brain. Now, there are some exceptions. There is a beetle that lays its eggs in charcoal, that is, in burnt wood. And so when it smells smoke, it goes towards the fire, not away from it like all the other animals. And the final reason why you might want to be able to smell, and at least in a human, is because you want to taste properly. And this is where we're going to do the little experiment. It's silly, don't you? If you should have a bonbon, a little sweet with you. Yeah, because you taste with your nose. I'm going to show you, right? And this is why it's so significant. The COVID-19 has revealed this to many, many people who, when they've lost their sense of smell through the infection, have also lost their sense of taste. So you should have, I hope, some kind of draguet or other bonbon. And what you're going to do when I tell you now is you're going to hold your nose, you're going to put the bonbon into your mouth, and you are going to chew it with your mouth shut just for about 10 seconds until I tell you to take your fingers off, and then you'll see what happens. Okay? So we're all going to do this together. Put your fingers on your nose, on passe le nez, on prend le bonbon, on met dans la bouche, et puis faut manger la bouche fermée. Alors, you can probably only taste not much. Take your fingers off your nose. On lâche les doigts. Et normalement, you should now get the full flavor. Initially, you will just have had a kind of sweet taste. But once you get the smells involved, then suddenly you get the full flavor. And this little demonstration, it's very bad to do for me because I get bits in my mouth. But I hope you've seen that works. If it didn't work, try it again later with another sweet or some food. Now, the reason why this has worked is that when we smell, we're not smelling here. We're smelling up. I mean, this still amazes me. We're actually smelling high up in our nasal cavity. It's about the eye level. That's where the actual immediate reception of smells takes place. And you can see here. So here's the eye, and the cells that are doing the detecting are dangling down through the bottom of your skull. So this is in fact part of your brain that is in contact with the outside world. And when we smell, what we've just done is, because you've blocked off the nose here, you've chewed here, and all the volatiles have accumulated, but there's no movement across your olfactory epithelium, this layer of cells that has where the detection is taking place. So when you take your fingers off your nose, then all those volatile odors that are built up in your mouth whoosh out across your olfactory epithelium, and suddenly you get the full effect of flavor. So this shows that we can dissect out three different things. There is smell, there is taste, and then there's the combination of the two, which is what we normally experience of flavor. And a huge component part of flavor is in fact from the smells that are released from the food as we eat it. And this tells you two things. Firstly, it explains why the COVID pandemic has had such an effect on people's sensory experience, because the infection takes place up here and it alters our perception of flavor. Secondly, it also tells us why old people often complain that food doesn't taste of much. And one of the reasons for this is that their sense of smell is declining, and they therefore have a much lower detection of the range of potential odors that are in their food, and they simply taste the basic kind of 5 or 6 sensory modalities associated with taste. So you smell with your brain, you taste with your nose, and you're actually smelling with your brain, these parts of your brain that are dangling down into your into your nasal cavity. And people often think that we don't have a very acute sense of smell. Normally I do this in person, and I demonstrate to the audience that in fact they have an atomic nose. Our noses are sufficiently susceptible to be able to detect single atoms in between two odors. We can see this first in something very similar like CARRAWAY and SPEARMINT, which have identical chemical formulae, but they are like optical isomers. Okay, so they're like two hands in a glove. You can see here that they have the same structure, but they are oriented in different ways. And these two odors smell very different, but they also smell a bit the same. And the more you think about those two odors, so CARRAWAY is one of the components of curry, and SPEARMINT, which is like mint. And clearly there's a hot cold dimension, which is interesting, although but there's also this similarity and a difference, and that is all coming simply from our ability of our sensory organs to be able to detect these chemical structures. We can equally see this in this example, which again normally I would give members of the audience these odors to smell. Very few people can identify these two alcohols, heptanol, which has got 7 carbons in it, and octanol, which has got 8 carbons. But people can detect the difference. They can see, they can smell. There's a very subtle difference. This one, people normally, it says it smells of violets, it smells sweet or woody. And octanol, people often say it smells sweet or orange. Octanol is often put in washing up liquids that smell orangey. They have octanol or nonanol in them because that has a slightly orangey smell. So you can see there's something similar because these are short chain alcohols, but there's also a difference, and that difference is a single atom of carbon. I think that's absolutely extraordinary that for animals that are generally think they can't smell very well, we can in fact detect single atoms of carbon. Now, in the industry, people often say that humans can, a good nose in the perfume industry can detect maybe 20,000 odors. Now, there's no actual basis for that. And recently, in the last 5 years or so, scientists have got very interested in, well, what is the upper limit to the number of smells we can detect? And the simple answer is we don't know, but at least one model proposed by researchers at Rockefeller University is that we can detect over 1 trillion odors. Now, that's a model. You can't give somebody 1 trillion odors and say, can you tell them all apart? But this is the kind of order of magnitude. I think effectively there is no limit to the number of odors that we are able to detect. Now, to give you an example, and many of you will know this kind of thing, that a rose can have up to 250 different molecules in its bouquet. Now, that doesn't mean to say when we smell a particular rose that we are detecting all 250 different molecules, different kinds of molecule, but it gives us some idea of the richness of fragrance in the natural world, and perhaps an idea of why we might need this incredibly sensitive olfactory system that we have. So how does it work? Well, like most things in science, to get an idea of how it might work, we need to go back to the ancient Greeks. And Democritus was one of the first atomists. So he came up with the idea that the world was made of atoms, tiny particles that he couldn't see. His head was made of atoms, the sea was made of atoms, everything. And he then thought, well, okay, in that case, smells must be made of atoms too. And what he decided was that nice smells must have round atoms, and horrible acrid smells must have kind of pointy shaped atoms. And that's basically where we are today. So in some way that we don't understand, there is a link between the structure of a molecule and how we perceive it. Now, the big breakthrough in trying to understand this came in 1991 when Linda Buck and Richard Axel at Columbia University in New York, they first identified the receptors, or they identified genes in a rat that they thought, and they were right, they thought those genes encoded the receptors. So the receptors are, you can think of them as like a lock, okay, in serrure. So you've got the key, which is the smell, and it comes in and it turns the lock, and then that enables the sensory cell, the neuron, to fire. They discovered the molecular basis of this phenomenon, and for that they were awarded the Nobel Prize in 2004. And the whole world of olfaction has changed utterly since that discovery. I was involved from the mid-1980s onwards, and it was like we were blindfolded. We didn't know where we were going. This discovery changed everything and led to a huge explosion of interest in the sense of smell. So we can now look at things in a bit more detail. So what's happening is each of our sensory cells, we have about 4 million. That number, I've no idea if it's true. It's something like that. We have around about 4 million olfactory receptor cells. So these cells that are dangling down up here. You can see here's the eye, the olfactory bulb, and each one of these cells only expresses one receptor type. We have about 400 receptor genes. We have 2 copies of each of those genes, and those genes can differ between each version. So you have potentially 800, normally around about 400, but potentially 800 different types of cell that is doing the detecting. Okay, and these all of the receptors expressing the same kind of molecule on the outside, receptor molecule, all of the cells go to the same place in the brain. These are structures called glomeruli. They're just the other side of the base of your skull, and they do the initial processing. So you're going to say, well, wait a minute, you've got 400 types of receptor. How do we get to 1 trillion smells? That's nonsense. Surely we can only smell 400 smells. Not at all. This is the kind of model that people have developed. The trick is that in the vast majority of cases, each receptor can detect more than one kind of smell, and each smell can activate more than one kind of receptor. So you've got an incredibly complicated code at the periphery. Before the brain gets involved, just in those cells here, there is an amazing level of complexity taking place. So here's an example. This is nonsense, right? This is not true. This is just a little model. So here we've got, say, 14 different kinds of receptor. You've got 400 kinds. And we can see here that in this model, heptanol, which we saw earlier on, is detected by receptor 2 and 6, whereas octanol is detected by 4, 5, 7, and 10. So you can see there's an overlap. They are both, receptor 5 detects both of these odors. So that's what's similar, but there are also differences. So you can see that by this combinatorial code, you've got effectively an infinite capacity of detecting different odors in the periphery and then doing various things with them depending on what you want to do. But as I say, what we don't know, we still can't predict from a receptor structure what kind of odor it detects. That is still beyond us. I suspect that in the next few years, we will crack that code. Research is beginning to do that. New methods of computer analysis of protein structure are enabling us to identify protein structure from a genetic sequence. So I think this will be resolved very soon. But does that really explain anything? How does it really work? And the answer to that, as I've indicated, is we don't know. So we don't know why one odor activates one receptor, but not another. So how can we find out? This is the thing that I'm interested in. I've given you the general background, but the actual detail, what's happening on a cellular level, how do those cells produce that image in our heads? That is much trickier. And that's what I've been interested in. And this is what I've been studying. So this is the maggot, l'astico, of a particular fly. And this is in its natural habitat. It lives in rotting fruit. This is a peach, and you can see this is the head, and these are the body, and that's the rear end. And there are lots and lots of these maggots in this fruit. And as some of you will have guessed, this isn't just any fly. This is la Drosophila. This is Drosophila melanogaster. And this is a male fly. And this is the adult. But I study the larval stage, so the baby fly. The problem with these animals, they are fantastic. Geneticists have studied them for a century. We know everything about them, but they are very, very, very small. So this is a USB key, and that's a Drosophila. So if you don't know how big, they're the ones you find around your fruit bowl because they like the smell of alcohol, because they're looking for yeast. So the downside is they're very, very small. And that's what a fly looks like. This is what a maggot looks like. This is the front end of a maggot. And these things here, they're not its eyes. These are what it smells with. These are its antennae, if you wish. Okay. So why would I want to study a Drosophila maggot when it's so small? Well, as I say, you've got around about 4 million smell cells grouped into 400 different types, but a maggot has just 21 smell cells, and each of them is different. Furthermore, maggots, unlike humans, are very stupid. They always do the same thing. They move along, they wriggle along, they can rear to side, move to side, but basically if they find, they're always hungry. So if they find a smell, they want to go towards it to find food. So they give very reliable behavioral responses. You can tell what smells they like, what smells they don't like. They are really ideal. They're very, very simple. And because this is Drosophila and we can do things with the genetics, I can make a maggot with just one smell cell in its nose. So this is the normal situation. And what they're doing here, they're glowing green because we are making those cells express what's called green fluorescent protein, which is from a jellyfish. So these are kind of transgenic Drosophila that we're studying here. And normally you've got 21 smell cells. Here you can see them all going up. And this is the nose, that little blob we saw on the outside. And this is a maggot with just one smell cell in its nose. So we can then record the activity of that cell and know exactly what it does when we stimulate it with a particular odor. So here's an example, and this will again confirm the incredible complexity of the initial processing of the olfactory signal. So here we've got two different receptors. OR means olfactory receptor, and this is 24A, the names don't matter, and this is 74A. So we've got two different single cells, and we're giving them three different odors, and these marks here simply mean the activity of the cell as it responds. The black bar is when we blew the odor, so it's a 1 second odor blown over the maggot's nose, and we can then record. And you can see here that butanol, which is a short chain alcohol, doesn't cause any change in 24A, but it makes 74A get very, very excited. So the same odor is producing no response or a very strong response. Octanol, in this case, actually stops the neuron from firing in 24A. So you've got inhibition. So sensory neurons fire all the time because that's just what they do. But this odor here is stopping the neuron from firing. On the other hand, if you give octanol to 74A, this other receptor, you get a very, very strong response. So it's not binary. You've got activation, you've got inhibition, you've got no change. You've even got changes in time. The behavior changes during the time and afterwards that you're being stimulated. So in the case of this odor, the neuron fires very, very strongly and then immediately shuts off. So you've got strong activation and then nothing. In the case of this neuron, you've got a double response. It fires and then it fires again. So I've just given you some examples there. What I want to show, the point of this is that each combination of odor and cell type will produce a unique response. This work has been confirmed by many other researchers. And what the brain is seeing is a change in spontaneous activity, which can be up, which can be down, or which can differ in time. Okay, so the response at the periphery is not like a switch, a 0 or a 1. It is amazingly complicated. Okay, what about humans? Well, we can actually get at the same kind of question using humans, but for the moment, just with one odor. So, as I said, in general, each odor activates more than one kind of receptor. So if you give a human an odor, lots and lots of our receptors will be activated. This is an exception. It's called androsterone. And as you can see, it's sold in sex shops for attracting women fast, but it also allegedly attracts men fast. In fact, it doesn't. It's a pig pheromone. And this pig pheromone is used by pig farmers to bring the sow on heat. It's normally produced by the boar, the male pig, in its saliva, and this will activate the female's ovaries. Now, what's unusual about this is that in humans, it is detected by one receptor only, OR7D4. The name doesn't matter. So we all have this receptor. It's present in everybody alive. And this is a little drawing of what the molecule is sitting on in the cell membrane. So you've got your cell and the molecule is sitting here and it's able to detect, it will respond to the odor. Now, what's interesting is that people differ very, very much in their response to those odor. And there are also genetic differences. We can see here, there are two genetic differences in particular on the outside and the inside of the molecule, which change how we perceive androsterone. So if you have these two, if you have an M, an R, these are the amino acids that are produced by these genes. We see it's single letters in the DNA are changing. So people like me who have a T here and a C here, so they have the amino acids M and W here, we think it smells nice. People who have a C here and a T here, they think it smells really nasty. So we can go from the genotype. If we see somebody's genotype, we can predict you will or will not like this odor. And we can also go the other way. If we know what somebody thinks about an odor, we can predict their genotype. This is very, very rare. There is this very tight link because OR7D4 only detects androsterone and similar compounds, and there's nothing else that will activate it. So I'm going to talk to you very briefly about some work I've done with some colleagues in America, Kara Hoover at Alaska and Hiro Matsunami at Duke University. And the first thing we did was to look at the distribution of these variants, these genetic variants in human populations around the world. These are indigenous populations. So they're not people like me that are all mixed up, but they're peoples who've been living in the same place for a long, long time. And just look for the moment at the distribution of the blue and the green and the red. So the blue on the one hand are people like me who think it's not so bad. Yeah, it's quite nice, sweet. And the people who really think it's horrible are the people in particular from Africa. Now, Africa is where we come from. So you would expect this to be the ancestral form. So originally we hated this smell. And then at some point a mutation arose, which you can see in the blue, which gradually spread around the world. And what's significant, and we have no proof for this, but we think this is our hypothesis, our data suggest that this mutation arose around about 5,000 to 6,000 years ago in Southeast Asia. And that is also the time and the place when we domesticated the pig. And if there are any pig farmers here, they will know that when you get a boar, a male pig is born, you castrate it immediately. Not only because if it grows into a boar, it will be very aggressive, but above all, because the pheromone, this androsterone that it produces, will taint the meat. It will make the meat taste of androsterone, which if you don't like it, is horrible. So we think that there is a link between this particular mutation and the consumption of pork. Now, we were doing this work, and then I had what is probably the only really good idea in science I've ever had. And I was out for a run by the river near where I live in Manchester, and I was thinking about this experiment, and I suddenly stopped because I realized we could use these facts to ask about our cousins, the Neanderthals. So the Neanderthals are extinct people. Their status has been uncertain, but what we now know is that we mated with them. And around about 4% of my genes, for example, come from the Neanderthals. So about 50,000 years ago, when we moved from Africa up into the Middle East, we met the Neanderthals, we mated with them, we exchanged our genes, and you can reconstruct about half of the Neanderthal genome from looking at modern humans. So they are very closely related. I think we're the same species because we could mate and leave hybrids like me and probably you. So I think that means we're the same species, but people argue about this. So we can look, we have their genome. People have been able to identify in frozen remnants of Neanderthals or found in very cold places where the DNA is still intact. You can extract the DNA, reconstruct their whole genome. So remember what I said. If you have the genome, if you have the genotype, you can predict what people respond to that smell. So it gets even better. Some of you may not know, you'll have heard of Neanderthals, but there are also another people called the Denisovans, about whom not much is known at all. We have a handful of teeth. One has just recently been discovered down in Laos this week. We have teeth. And from these teeth and a small finger bone from a girl, which was found up here in a place called Denisova in Russia. And these people are called the Denisovans. We don't have any, they're not Homo whatever. They're just the Denisovans for the moment. We don't have enough of their bones. But from the from the finger bone that was discovered in 2010, they were able to sequence the genome and discover that there were this whole previously unknown group of humans who, again, we mated with. And perhaps 8% of the DNA of people, of Australian Aboriginals, for example, comes from the Denisovans. So when we expanded out of Africa, we met the Neanderthals. Then as we went through Asia, up into China, we also met the Denisovans. We mated with them as well. We exchanged our genes. And these are extremely important discoveries for biology. But what we wanted to know is, what would the Denisovans and the Neanderthals have thought about androsterone? So we can look in their genome and see what variants they had to try and understand. And what we found was that the Neanderthals had the same version as modern African populations. And that's kind of what we expected, because the African version is the original one. The the Neanderthals left Africa first, 500,000 years before us. So they had that gene. They would have hated it. You would have given it to them and they would go, oh, it's disgusting, disgusting. Now, we got very excited because the Denisovans had a novel variant, one that we haven't seen in any human. So we've looked at thousands and thousands of sequences, but this was completely novel. So what was this like? This was a very exciting moment. Well, it was before we got the result. So what we did was to mutate a human receptor. We mutated the human gene in a cell line, not in a person, in a cell line, changed it to have this single base pair change. And then we put some androsterone on the cell And sadly, it didn't change the result. So what we think is that the Denisovans also, we expect, would have hated it. Now, it's only really possible to do this with androsterone. However, we have recently, we have a paper that is currently being reviewed in which we look at about another 10, 20 receptors and the odors that they're activated by to look at how androstenols, how Neanderthals and Denisovans would have responded to other odors. And the very short answer is that Neanderthals probably quite liked sulfurous smells, so farty smell, l'odeur d'épée, and Denisovans probably quite liked sweet smells, so the smell of honey and so on. So this may tell us something about their different ecologies, because the shape of our olfactory receptors is altered by natural selection. So this is going to be a way of understanding not only the perception of long dead peoples, but perhaps also give us insight into their ecology. So what can you take from this? One, we still don't understand how the sense of smell works. The way that the nose interprets smells is very, very complicated. As we now know, loss of olfaction can really affect the quality of life, and you can do something about it. So there are various ways of doing smell training that can improve when you have lost your sense of smell through injury or disease. We've got this astonishing power of using genomics to look into the deep past. I think this is the most amazing thing. And of course, as always in science, it's what we don't know that's really interesting. Some of this, a lot of this is explained in this little book, Smell, which has recently been translated into French, L'odorat. Here you are. It's very thin. And that's your lot. Thank you.
Well, thank you very much, Matthew. Smell is a fascinating world. I see I have one question. I tend to limit the question. I'm sorry, but we have some delay, and the other speaker have some constraint in terms of timing. So time for the first question, and it could be the only question. Excuse.
Do I have to do this or?
Excuse me.
Do I do this or I can see some of the chat? I can read them out if you want.
So we have a problem. I am going to read the question.
Okay.
How do you remove deodorants during stimulation of maggots' sensory neurons?
Well, you have a continuous flow of air, and you briefly, you have a valve which releases the odor just for 1 second. But I mean, this is very artificial. So no, this is what everybody does, whether you're studying rats or humans or maggots. You give a brief pulse to try and understand what the neuron is doing. But a lot of us think the reason we do this is to control, to limit the variability. But if you think about it, that's not real. Our real sensory world involves, for example, detecting odors against a background. So if you smell perfume or food, there are lots of other smells going on around you. You're not simply smelling that one odor for that brief second. And the understanding how blends are detected, and virtually all odors are in fact blends, that is a huge area of research and of debate, and people get very cross with each other because there are different models that we haven't yet resolved. So what I've shown you is true, but it is also extremely artificial, and the real world is very, very complicated. The world in which animals and humans have evolved to have a sense of smell involves long periods of odors, adaptation to those odors, and then detecting an odor against a background, and above all, detecting blends. And that is the big challenge for olfactory science, I think.
Okay, thank you very much, Matthew. And it is true, we have a fascinating book to read quickly.
Okay, thank you.
So I am sorry because we are in direct, and we have a timing constraint, so notably with Valentina Parma. So I propose you to modify the order of lectures. And Valentina, which is based in Philadelphia and which was kind to wake up very soon this morning to participate to this webinar, is going to present you the result of our studies about sensory effects of COVID-19. So, Valentina, it's up to you.
Thank you very much. Thank you very much, and thank you for adjusting the schedule for me. So I'm Valentina Parma. I'm a psychologist by training, and I'm currently the assistant director of the Monell Chemical Senses Center based in Philadelphia. And Monell is a center that is devoted to the study of smell and taste in all its different facets. And so, as you can imagine, during COVID-19, we've been quite busy, because as you all know, smell and taste became some of the specific symptoms of these disorders. And so today I want to walk you through the sensory effects of COVID-19, like what happens to the nose, and to kind of walk through what happens after these type of sensory issues appeared. And so we're going to start by looking at what happens exactly in our organs of sense, that as Matthew very well presented before, what happens exactly with the molecules, where exactly it happens. And so where are the receptors in the nose and the mouth, and what is the result of COVID-19 on those. We're going to then focus on what happens at the perceptual level. So when the brain integrates all of this sensory information, what is the result? What people are telling us about their experience? Are they able to tell us anything meaningful that we can, connect with and then adjust to? And then we're going to look at behaviors. And so in particular, one of the majority of the reports that we hear about is how, the ability to eat and experience the smells and the flavors in the context of eating has changed for people that had modifications of smell and taste due to COVID-19. And then we're going to also take a long-term view, because, for most people, and, this is a spoiler alert, but for most people, smell and taste changes are going to be quite temporary. But what happens when they're not? And what are the long-term consequences on our body weight, for instance, body composition, or even our mental health, which is related to our ability to eat and enjoy food? And so COVID-19, as you may have heard, really threw a fit on all of these systems at every single level. And so today I want to kind of review with you, these steps one by one. And we start with the receptors per se. And so SARS-CoV-2, which is the virus that, provides the disease that is called COVID-19, impairs the biological substrate of both smell and taste. And we have quite a lot of information about this happens in smell, although not all mechanisms have been mapped. And if you think about, I'll bring you back with me to March 2020, when, myself and others were receiving a lot of messages on social media from people saying, I got COVID, my smell, I cannot smell anything. What happened? My food tastes weird. And so one of the first studies by the group of Bob Data at Harvard and David Brand is the first author of that study. What happens there is that they looked at in animals, how was the distribution of certain receptors? And the hypothesis at that point was that SARS-CoV-2 would bind to olfactory sensory neurons and kill them. But this is not quite what happens. So when the virus enters the nose orthonasally, and then it reaches the level of the olfactory epithelium, which and I'm gonna use my pointer here, at the level of the olfactory epithelium here, where the receptors are, what happens is that we thought, those olfactory sensory neurons would be killed. And so the information after that would not go into the olfactory bulb, it would not be passed into the brain. And so that would be the reason why we're not able to experience smells, is simply because the whole system, got cracked from the beginning. But then we started, this is not what the group by Bob Data suggested. It's not in olfactory sensory neurons, which was the hypothesis of most scientists at the time, but it's SARS-CoV-2 attacks the cells that are supporting the life of olfactory sensory neurons. These cells here, which are called supporting cells, that are feeding the neuron and keeping it alive. So if SARS-CoV-2 attacks the supporting cells and kills those cells, as a result, the sensory neurons are not working. But there's not a direct, elimination of olfactory sensory neurons. And if we want to use a metaphor that Federica Genovese, who's a colleague of mine at Monell and a neurobiologist, uses, you imagine SARS-CoV-2 as kind of, somebody punching on a keyboard, and where you have all the keys going everywhere. The olfactory epithelium is not in the shape and in the order that it used to be before. And as a result, perception is very much changed. And it's changed primarily in two main ways. So we've all heard about smell loss. And so we all heard about people, and maybe some of us have experienced that, thinking that, okay, I got COVID, my smell, disappeared completely. And as you can see here, I have all of this like keys that are completely disorganized. Like I cannot smell a thing. But then perhaps after 2-3 weeks, maybe after a month, or maybe after 2 months, people start experiencing a different type of smell disorder. And so they start experiencing some smells, but those smells are not quite exactly the smells that they know were connected and associated with a certain object. And so if you think about the odor of an orange here in the middle, and you have anosmia, you cannot say that this is the odor, that there's an odor at all. But if you have anosmia, you may likely say that there's the odor of trash rather than the odor of orange. And you're quite surprised. And so if you go back to the idea of, the keyboard and this metaphor, you kind of have all the keys that you need to build the orange smell, but they're disconnected. They're not in the right order. They're not placed as they used to be, as you can see here. And so you have a distorted experience of what the smell of orange is like. And we're describing this as, quite an objective, situation. But yeah, it's very, very disturbing because imagine the world not being exactly as you imagine it to be, or you learned it to be for a long time. And so what we know, and from people that are experiencing this condition, is that it's very, very disturbing. And as I mentioned, I'm mostly focusing this talk on the nose, but I just want to give you also a hint of what happens into the taste system, because as we've seen before, thank you to Matthew's talk with the candy experiment, these experiences are extremely intertwined. And in SARS-CoV-2, we assumed, again, as scientists, that people, lost primarily their sense of smell. We had a mechanism that could explain that through through the supporting cells misfunction. But what happens to taste? The people are reporting that taste that doesn't feel exactly the same, but usually we know there is a phenomenon called the smell-taste confusion, based on which people attribute to taste the things that are really coming from the sense of smell. But in SARS-CoV-2, we're gathering more and more evidence of the fact that taste is actually impaired in and of itself. And so we do have pure taste loss or distortions, which is before COVID-19, it was quite a rare occurrence. And so we know that the more SARS-CoV-2 we find in saliva, the more taste loss people tend to report. And this is still a rather controversial finding, but more and more studies are going in that direction. And if we look at the immunohistochemical staining on the tongue, we can actually see that there are a reduced number of taste buds in presence of SARS-CoV-2. And so as you can see, really, COVID-19 and SARS-CoV-2 really affects this biological substrates. And so what happens to the perception? I told you a little bit about anosmia, I told you a little bit about parosmia, but there is a little more than that. Sudden loss of all three chemosensory modalities is what is reported in COVID-19 people, in particular as compared to people that have other types of respiratory disorders like the cold or the flu. And how do we know that? Well, we do know that in particular from the research of the GCCR, which is the Global Consortium for Chemosensory Research. And here you see approximately 700 people distributed really worldwide that got together at the beginning of March 2020 to answer these questions like what's going on with smell and taste, and even chemesthesis like oral and nasal irritation with COVID-19. We wanted to make sense, and we, with our research, help certain groups and governments recognize that they needed to include smell and taste loss and changes in the list of their symptoms for COVID-19. And this is the specific case of the UK in particular, through the efforts of Barry Smith, Chris Kelly, and Claire Hopkins. So as you can see here, we do have two major publications, and I'm glad to report that also Charlotte was part of this super co-author group, based on which we could really detect very early what happens in smell and taste and oral irritation with COVID-19 patients, and with COVID-19 patients compared to people that have respiratory disorders. And what do we find? So here we have 4 panels, and all of these refer to one chemosensory modality. So if you can still see my my pointer, in purple we have smell, in red we have taste, in yellow we have chemesthesis, and then we have this pink which is nasal blockage. And so as you can see, the left side part of the graph refers to reports from people of their experiences with each of these senses, or experiences in general before COVID-19, and then what happens during the disease. And as you can see, things are all dropping. There's a complete loss of minus 80 points on a scale from 0 to 100 for smell loss. So there is a severe taste loss, a change in 70 points. And then we do have a variability in chemesthesis loss, with some people losing a lot of it and some people not reporting any change. But what is striking here is that people tend to not report nasal obstruction, meaning that people do not report a stuffy nose while they lose the sense of smell. And this was quite surprising, because it really it was a breach in our expectation, in the sense that the majority of people that are reporting smell loss following an infection, a viral infection, they tend to report it like 2 weeks, 3 weeks after the infection has cleared out, because concurrently they tend to have like a stuffy nose and they do not detect the changes in their sense of smell. But with COVID, it was really different. We had participants in our studies describing that they were sipping their coffee in the morning. First sip, everything was fine. Second sip, something started to change, and third sip, they couldn't really smell their coffee anymore. And so it's quite a striking mechanism. And when we in the summer of 2020, we really wanted to answer this question of, can we detect anything in the chemical senses that can help us discriminate whether this is COVID or the flu? Because we do know that the symptoms are primarily, shortness of breath and coughing and some cold and sneezing, but this is very common, not particularly specific to COVID per se. And so could we actually use smell and taste to discriminate these two groups? And I was in contact with some emergency groups at the hospital, trying to figure out, they were very interested in having a test to figure out whether smell was impacted more than the general flu. And so as you can see here, again, the first graph is smell, the orange is taste, the green is chemesthesis, and then we have blocked nose again in brown. And when we compare people with a COVID-19 positive test versus people that had respiratory symptoms but a COVID negative test, and be mindful that this was at the beginning of 2020, so this was the wild type of the variants that we were looking at, we can see that we find much more smell loss, taste loss, and chemesthesis loss in people with COVID-19 as compared to people with a respiratory disorder that is not COVID. But then, if you follow the literature and you kind of want to, have a more long-term outlook on these effects, because the majority of these studies were collected the data was collected very quickly through self-reports, because if we think back at the beginning of the pandemic, it was impossible to get into the lab and, have people smell things that were standardized. And so if they were not in a hospital. And so some of my colleagues at Monell, Mackenzie Hannum, and the group of Danny Reed decided to look at all of the studies that have been published in the up to March 2021. And so by pulling together all of these studies, we can actually have an idea of what is the general feeling, irrespective of methodology, irrespective of the sample. What are we actually finding? And so if we look at smell and we ask questions about your smell function in two ways, that we can ask, can you self-report how good or bad your smell is? And so people are just not smelling anything, but they tend to respond based on memory. Or we have an objective form of testing. So we have people either smelling some household items or a standardized test. And as you can see here from this graph, we actually collected cumulatively a bunch of people, like 66,000, 3,000, 2,000. So these are large groups that, analyzed together can help us understand a little bit what are the trends. And so what we see here is that when we look at smell testing, we detect 69% of people that have lost their sense of smell with an objective smell test versus only 47 if people report, telling us that 22% of people do not notice whether they lost their sense of smell, give or take, if they're not faced with an olfactory challenge themselves. And there's a different situation here that we taste, because we can see that the subjective information and objective information are quite reliable, and approximately 40% of people, irrespective of the method that they're using, would report some sort of taste loss. And by taste loss, I really mean not loss in flavor generally of a food, but I really mean a change in what is the perception of salt, of sweet, of sour, of bitter, and of umami, like very, very precisely asked. Interestingly, too, there is also chemesthesis loss, as I mentioned before, and I think this is the best case that I've seen. There is this is a neurologist from the UK who, in the very early days of COVID, got COVID himself, and he videotaped himself while eating a chili pepper, extremely spicy. And this was the first video, and it was just, I can entirely eat this chili pepper, super spicy. I feel nothing, and he kept like eating. Thankfully for him, 2 weeks later, he was actually able to redo the same test when the chemesthesis loss vanished, and he was really crying like a baby, indicating that there was really a marked and profound change in the ability of experience trigeminal sensations orally. And as we've seen before with the meta-analysis slide, so with the cumulative study, we can see that we're good at catching cases with direct smell test, but not with taste testing. And so how can we actually improve this? And this was a big problem, because at the beginning of the pandemic, like again, I mentioned before the ER departments, they were using alcohol or some of the, the materials, the olfactory materials that they had in hand, but usually they tend to have a trigeminal component, which is, as you've seen, like not everybody is losing their ability to perceive pungent odors. Therefore, we're really underestimating the people that are having smell loss. And the tests that we have that are already commercialized, they're either too long, too costly, or they are prone to contamination. And so we're really preventing like using a smell measure in the clinic, in the workplace, or at home to monitor changes in one's smell and taste. And this is why we tried at Monell with Pamela Dalton, we tried to figure out how can we actually provide a rapid smell test that meets the scientific needs. So we're not only asking about odor identification, for instance, because we know that is particularly biased and it is culturally dependent, and therefore for a population level surveillance, this is not particularly helpful, and we need it to be practical for a COVID world. So it has to be fast. We cannot have people wait for 10 minutes to enter a building to complete a smell test. We don't need to use coins or pencils to scratch and sniff something, and we do not want a bin of cards disposed that give off all of the odors of the hundreds of people that have entered the building before you. And so what did we do? We developed a test which is called the Sentinel. That is on a card, and we have 3 labels. So the patches A, B, and C that you see, these are label odorants, and 2 of them present an odor and one doesn't. 2 of them present no odor and 1 doesn't. And as you can see here with the QR code, you can just use your phone to access the test and the questions, or you can just type in this address here onto your web browser, and you're able to receive different questions that are customized based on the type of test we want to do. But the core Sentinel test asks 3 questions. What is the odor that you're smelling? Where is the odor that you're smelling? And we're just find which one is it, A, B, or C. How strong is the odor from 0 to 100? And what is the odor? And I'll give you 4 options because that would be otherwise quite a hard task. And so at the beginning, in August 2020, we decided to connect with a group of people from the Monell mailing list. So we fully remotely were able to test people, and we identified 154 individuals with normosmia, and we gave them Sentinel, and we gave them also the NIH odor identification test, which is part of a standardized test, which is a gold standard in the United States, and it's a scratch and sniff odor identification test. We then identified 111 individuals with anosmia, and we asked them to complete Sentinel, and then we had 42 people that presented other types of smell disorders, hyposmia, parosmia, like a mix. And what we're able to say is that we really were able to discriminate people that have a normal sense of smell with people that completely lost their sense of smell quite accurately. And so this curve here represents like how good is the test to discriminate these 2 groups. And the more the curve goes onto this top left corner, the better the discrimination is. And as you can see, if we just use one of the subtests, we have 80% of ability of discriminate these 2 groups. If we look at odor identification, we have 84% of ability to discriminate normosmics from anosmic, but then we get up to 94% when we look at odor intensity. And when we put this course together, we really are able to only misrepresent 5% of people by using this test, which takes less than 2 minutes. So we were really, really happy. And when we look at the ability of Sentinel to compare to a standardized test that have been utilized on thousands of people, we actually see that we have similar results, but in a fraction of the time, like 3 times less time as compared to the NIH olfactory toolbox. So we were very, very pleased, and we started to think of how can we use not only this test for diagnosis and detecting those 22% of people that would not be able alone to detect the presence of smell loss, but also to start using this type of test to monitor recovery, because recovery is quite variable over time. And so to give you a few numbers here, if you have had some smell and taste loss, really do not feel alone. And if, and try to consider the magnitude of this phenomenon for everybody. So here what we have is that these are the number of people that have had COVID. It's even higher right now worldwide. If we think that 69% of people, give or take, have had at a certain point in time a COVID smell dysfunction, we're talking about 170 million people. And if we look at the fact that 10% of them tend to have long-lasting smell loss, we're talking about 17 million people. And this is like new people that in the past 2 years started having issues with smell loss. And we can have an analogous reasoning with taste, and we think about 40% of people. And, if we want to be very, very conservative, we have 15 million people worldwide with a new loss of smell and taste, or taste disorder that needs to be addressed. And so this requires a lot of attention, which is not what we currently find when we enter the clinical domain. So doctors may not know about smell and taste loss, and they do not know what to do. And so I see the questions in the chat, and I'm going to address them. What can we actually do to make this better? And if we start seeing, some of these 10% may actually be a conservative estimate. When we look at the data from GCCR, where people were already tuning in and interested in smell and taste, we actually see that the data are a little greater, and that people tend to present parosmia often, like these distortions of smell that I mentioned. Early recovery, based on our GCCR data, seems to suggest that we can recover, like more people in the first 30 days tend to recover at least 50%, and we did have 50% of sample that didn't yet recuperate their sense of smell. When we look at people that had the flu, these percentages are very different. There is a 30% of people that never lost their sense of smell against 9% of people for COVID-19. And we tend to have people that recuperate more than they don't. But what happens over time, what happens with later recovery, like more than 90 days after, and as you can see here, if you have contracted, like if you have preserved the kinesthesis, so if you have the ability to perceiving oral irritants, it seems to be more frequently associated with substantial recovery, indicating that you have had a spike in an inflammatory response, which once it goes away, allows the smell to come back as well. Those who recover faster tend to be younger and to have reported initial greater loss, and this may be just an effect of noticing larger differences in the experience. And as I mentioned, this is a very good representation of what parosmia feels like if you have not heard somebody talking about it. This is a drawing made by Christy Kelly, who's the founder of Absent, which is a charity in the UK that is devoted to helping people with smell and taste loss cope with this condition. And she suffered from brutal parosmia herself. And this is the drawing of what her experience is. Coffee is terrible, onion, meat, fried food, like all of these are extremely triggering odors for her. And as you can see from this like word cloud, for thousands of people that are part of her Absent groups on Facebook, we're talking about more than 10,000 people at this point. And what we can see is that there seems to be some personalization, like the tiny small world indicate that everybody has some idiosyncratic bad experiences, primarily bad experiences with odor distortions, but there are a few odors that are recurrent. And so there are scientists like Jane Parker in the UK that are trying to figure out what are the chemicals that are triggering parosmia and why. We still do not have a full understanding of the mechanism. And if we think about like this variety of olfactory experiences during the course of COVID, because as we said, we may not be able to catch it like early enough and very quickly, what we do see is that we want to see whether we can have a test that can detect at least these 4 groups. And so we're looking at people that have anosmia, complete loss of sense of smell, hyposmia, partial loss of sense of smell, parosmia, distortions, and they have a normal sense of smell. And so we use a sentinel here again. And we collaborated with Absent to make this happen. And we created a Sentinel Plus version, if you will, in the sense that we are adding 2 questions. So not only we ask where is the odor, how strong it is, and what it is, but we also want to know how pleasant it is. And we do so by using one test that has been used by Liu, who is a group, a scientist that is part of the larger group of Thomas Hummel, who is the leading world expert on this topic. And he is asking, imagine the smell of vomit, which most people would consider unpleasant, and we want to create a score, an hedonic score, with the difference between these 2 odors, the odors that you're smelling on the card and the imagined vomit odor. And as you can see here from these curves, again, the closer the curve it is to the top left corner, the better it is at discriminating these 2 groups. And so here we're collapsing people that have quantitative disorders, meaning that they lost some or completely their ability to smell, or they have distortions. And we can discriminate hyposmia from parosmia with 83% chance, or 92% between anosmia and parosmia, and 93% with anosmia and hyposmia. And so we conclude that Sentinel, it's actually the first direct test, an objective test that can allow us to discriminate these 4 categories in less than 2 minutes. And we're really proud of all the applications that this can have. And let's think a little bit broadly of what it means to lose your sense of smell long term. And we do have direct effects on nutrition. I mentioned many times that primarily people report changes in their ability to eat. But what does that mean exactly? And so here, when we look at the data, what we see is that, thinking about changes in the way you eat as a result of smell loss. It's not something that happened only with COVID. Here we have a study like from 2015, but really from the 19s, we kind of have the same data from people losing their sense of smell for whatever reason. And so 25% of people report that there is a decrement in the enjoyment of food, and there is a reduced quality of life, among other things, less social sexual behavior changes. I have professional difficulties, difficulty with cooking, safety issues. I cannot smell if something is burning in the house, and this is dangerous. And a survey of these 487 people with COVID-19 actually told us a different story. Like, I have less enjoyment for food. It's for 85% of people, not for 25%, and I have worse appetite before they became ill. So you can see that we have a much greater impact of enjoyment of food from COVID-19, and also in particular due to parosmia and long-term parosmia. And so if we put it in the words of the patients, we actually see that from a thematic analysis from the absent group that I have already mentioned, most participants described anosmia and the flavor changes as very problematic, and they changed the way they eat, meaning that they either eat more or they eat less, and food became like unappetizing or absolutely abhorrent. And in our data from GCCR directly, we actually identify exactly the same changes. So when people have changes in flavor and in smell, we can see that there tends to be loss of appetite. And I'm almost done. If I can finish this concept, is that okay?
Okay. Excuse me, Valentina. Could you a little bit accelerate, please, because
Yes.
we have great delay. So
I will finish this
fascinating,
concept.
but okay, thank you very
I'll
much.
finish this concept. And what I was saying is that we do have, people that lost their sense of smell tend to eat more, and people with parosmia really tend to eat less. And so if we are going to consider altogether what these different changes that we have seen, we see that COVID-19 really changes the substrate of our ability to detect the odors and process the odors. We do see that people tend to have like nutritional consequences that changes their way to eat in terms of increasing weight gain or weight loss. And we do see that the pandemic, like all of the millions of people that are showing smell and taste disorders, are not only they are accumulating other types of sensory loss, which usually happens when we get to 60 years of age, more or less. And so we see that altogether, smell and taste loss may actually significantly impact on what will happen in the coming decades for all of us in terms of nutrition, in terms of mental health, in terms of healthy aging. And so it is very much important to understand how to address these issues very early. Very quick plug-in acknowledgements to all of the teams that my research was based on, and I thank you for your question. And I can answer to the questions in the chat in the interest of time, if this is helpful.
Okay, thank you very much. I am terribly sorry to so speed up the presentation, which was very interesting. We have still a presentation. So are there some questions about this very important problem, notably for the professional of flavor and fragrance industry?
Would you like me to answer them live, or I can write the responses if it helps?
Yes. So perhaps I don't see question for the moment.
I will write those that I see in the chat.
I find the question. Excuse me. It's okay. I'm going to change.
Yes. So is there a correlation between severity of COVID-19 symptoms and capacity time of recovery of the olfactive capacity? So the data are a bit mixed, but as you've seen from what I presented, is that the people that tend to lose the most at the beginning tend also to share that they regain faster. And usually people regain their sense of smell within 2 weeks. And after a month, now we're considering that those symptoms are an indicator of long COVID. So there is not a clearly linear relationship, but there's a noticing type of bias. And in terms of the methods of recovery, since I see the other questions, there is only one recommended method at the moment, which has 30% chance to improve, which is called olfactory training. And it's basically requiring to smell odors repeatedly, multiple times a day, or at least twice a day for 5 minutes. And it seems to be helpful for 30% of people. The mechanism behind it's still not super clear. There are several, right now there has been an increase in research in trying to understand how to improve smell and taste loss. There is like a plasma-rich platelets seems to be an interesting method that has ongoing research trial. There is theophylline, but the data is still not very clear on this.
So we have some another question. I am going to publish them and to ask you if you can reply in 1 or 2 sentences, please.
Sure.
Valentina.
Yes. Yeah, recovery, we tend to recover in 2 weeks. The majority of people, 85% of people recover in 2 weeks, but more people, 10% of people takes more than a month at this point.
Methods to help recovering?
Yes, what I mentioned about olfactory training is what we have as the recommended treatment. And then we have experimental strategies that are being investigated right now. So the more prolonged the smell loss, the more complicated the recovery. That seems to be that seems to be the case. But again, recovery is non-linear, and I think what people start to think and clinicians start to think is that within 24 months, a lot can happen. After 2 years, the adaptation is a little more difficult, but the adjustments are still possible. We have cases of recovery of smell after 9 years, not due to COVID, of course.
We have already talked about that.
Yeah, the only recommended one is olfactory training, which is effective in a limited amount of people.
And the last one?
How can we recover smelling? And it seems that the olfactory training paired with cognitive abilities, like visually representing the odors and collecting memories about the odors based on the patients and those that have lived through these conditions, seem to be effective. But we do not have the highest clinical trials at the moment to confirm with the scientific perspective exactly how that works.
Okay, thank you very much, Valentina, and for your effort to stand up very soon this morning and this very interesting presentation. So I wish you an excellent day, and now it's time to talk about flavor perception with Charlotte Sinding of INRAE at Dijon. Charlotte, excuse me for the delay. I am an horrible time skipper.
All good for me. I hope people had time to have lunch during the presentation. So I will try to keep my talk short. I just would like to thank Matthew and Valentina for their great talk, because they also bring the idea that smell is very important for taste perception, and that's the topic of my talk today. So I will present you 2 studies, one on neuroscience of odor and taste integration. So this was done with electroencephalography. I will explain you that just in a moment. And the second study is about the odor-induced taste enhancement. Some odorants have the ability to induce taste perception. And I will present you a bunch of results about some of the odorants we tested. And we also look at these mechanisms into population in people with obesity and in normal weight population. So just to introduce very quickly where I come from. So I'm a researcher at the Science Center for Taste and Feeding Behavior in Dijon. You probably know, at least for the French people, that Dijon is famous for food and wine and taste. And this is important because that's the reason why our lab has been built in this place, and it gather different institutes from French Institute, INRAE, CNRS, Institut Agro-Dijon, and the University of Burgundy. And we are 10 teams working on food perception, food behaviors, from the molecules until to the brain perception, and with different models. So I go to the main aspect of my talk, that is the flavor. So the flavor I define it as a unified brain representation of food that necessitates at least the integration of odor and taste perception. So we saw with Matthew that odor and taste were often mixed, and that this is due to this integration and the creation of the flavor by the brain. So what we know is that odor and taste have different receptors that are located on different sensory organs, and they also have different cortex. So the areas are very well separated. Nevertheless, we know that odor and taste at some point are integrated. So we know part of the flavor network, but we don't know there are many areas that participate into the flavor perception, and we don't really know which one will really contribute to the formation of the flavor and how this network is organized. So that was one of the questions we addressed with electroencephalography. Just before presenting you the result, I show you that many studies show this odor and taste areas. So some brain areas are responding to odor and taste. This was done by the lab from Small in the US, and they did a meta-analysis of fMRI data. So fMRI is good to locate the brain areas that are involved in certain type of stimulations. So they gathered data from fMRI studies on olfaction, some other from taste studies, and they show that some of the areas are overlapping between the two perceptions. So these areas could be areas of flavor formation. And then they showed that within this set of areas, some of them were showing a certain response, so more activity in response to odor and taste when they were mixed than when they were presented separately. So they tested vanilla and sweet perception versus vanilla and salty, something incongruent, at least for the participants who were involved in the study. And they tested also the 3 separate ingredients. And what we see is that there is a response in some of the areas that is much higher for the congruent vanilla sweet mixture compared to the components presented separately. So it seems that there is integration. This result is really the demonstration of the integration by the brain. So if we look at the literature, there are a bunch of studies that shows actually this type of results, so supra-additive response, super-additive activation for odor and taste mixtures compared to odor and taste presented separately. What we see is that there are mostly high-level cortices, so brain areas that will process odor and taste after it was processed by very primary sensory areas. And it is relevant because these areas are known to be integrative. So they can integrate odor and taste, but we know that they can also integrate some other perceptions, also emotions, and so on. But interestingly, there were also some primary cortices that show these supra-adaptive responses, although it was unexpected. So in these fMRI studies, a lot of them show that the primary gustatory cortex was activated more for the mixture of odor and taste. So the anterior insula and frontal operculum. And this was shown only in animals. It was never shown in human, but it seemed that the olfactory cortex is able to respond to taste stimulation. So the piriform cortex in this study from Meyer was shown responding to taste. So our question was actually, where is the flavor perception created and when, at which moment within this network? Because if this is happening in the primary cortices, it's a very short response. These are the primary areas activated. And it involves less integration from emotions, memory, and so on. So there are different implications. So these are the two views about how odor and taste are integrated. And there is still a debate on which one is actually the relevant one. So on the classical and older view is flavor is created in higher order area, orbitofrontal cortex and amygdala, and there are no communication between the primary sensory areas, which is for taste insula and for smell piriform cortex. In the more recent view, with the results I presented to you today, in the literature that's just presented before, we saw that there could be activation and communication between primary sensory areas. So we wanted to test that in human, and we use EEG. So I will show you how we did that just afterward. But I wanted to introduce also the idea of the event-related potential that is the result we obtained from electroencephalography. So we put electrodes on the scalp of the participants with this cap, and participants are stimulated with odor, taste, or the separate ingredients, and we get this potential. So this is the beginning of the stimulation, which lasts between 200 or 400 milliseconds, depending on the studies. And what we obtain is this potential with peaks that vary from up to down. And these peaks represent moments where a large group of neurons will synchronize to process the stimulation that was given to the participants. So we can put on this scheme the views I presented to you before, because actually these peaks corresponds to the different moment of the processing of the odor and taste, and they correspond to these different brain areas. So we can apply on this graph that this first peak will actually be connected or will represent the activity from the primary sensory areas, so the early processing of the odor and taste, while this later peak will correspond to more higher brain areas, cognitive areas, and integratory areas. So we can imagine that if we see modification on this late peak, then and no modification on the first primary sensory peak, then the integration of odor and taste will take place in this later peak. So that was one of our hypotheses, and to show that we used a phenomenon that is the odor-induced taste enhancement. Because with this phenomenon, we can be sure that the flavor network has been activated completely. So that's a sensory test that allows us to know that the flavor was processed and integrated. So this phenomenon can be explained in this way, that when we receive the salt on the tongue, there is a brain activation with a dedicated cortex, and we have a salty intensity. And what we see is that when we add, for example, a chicken roasted aroma, just the odor is perceived through the olfactory cortex. So this was explained by Matthew before. And what we found is that the salty intensity increased. And it seemed that it's not only an association, so a memory increase, but it seems to activate actually the gustatory areas that are involved in the taste intensity perception. So what was shown in sensory studies is that there are a bunch of aromas that can increase the salty perception. So here the participants were testing and evaluating the taste intensity of solution with a low amount of salt and with aromas added. And we can see that some of the odorants increase quite highly the taste intensity, while some of them do not increase the taste intensity. And this one is carrot, the aroma, the carrot aroma. It's even decreasing the taste intensity. So it seems to be aroma dependent. So this was shown on salty perception, but also on sweet and sour perception. So I come back to the EEG data and what we did to show this organization of the flavor network. So we use this electroencephalographic recording, and we used a gustometer that allowed to send odor and odorants and taste to the participants with a very controlled way. So we send sequence of stimulation that are very precisely resolved in time, so we can be at the resolution of neurons, firing neurons. So this is very helpful to do these studies. And we, as I explained to you, we look precisely at the P1 peak, so the one that is connected to the primary sensory areas, and the P3 peak that is connected to the more cognitive and high order areas. So we stimulated the participants during 400 milliseconds, and we had 40 repetitions of each solution. This is important to have a signal that is without noise, because of course when we are recording, the people are still thinking about what they are doing and also too many things, so the brain is still ongoing. But we want to look only at the brain signals that are linked to the stimulation. To do that, we have a very high time resolution that is linked to the stimulation, so we know exactly when the participant was stimulated. And all the brain activities that is not linked to this stimulation is removed by averaging the 40 repetitions. So this is very classical in neuroimaging studies, to repeat the stimulation to have a clear denoised signal. Okay, and this I will not go too far into that, but just participants were trained to have a velum opening, because the velum, as Matthew showed you, can block the flow of aroma into the nose. So we trained the participants to keep it open. And participants could not swallow during the recording, only at some point where we were not recording, just to avoid that there are muscle movement artifacts during the recording. Okay, we tested 3 different solutions, 4 actually. So the base solution was a complex food, so we wanted to be as close as possible to real food. So we used an unsalted green pea soup, and we used a usual salt level, the one that is normally found in this type of product, and we decreased it by 50%, but we also used the beef aroma to compensate for the saltiness intensity. So we stimulated the participants with the reduced solution, the ones with the reduced taste, the reduced salt level, plus the aroma, and the usual solution. And we use some controls. So we validated first that there was this odor-induced taste enhancement. So we can see with this solution that the intensity is increasing, not to a very high level. We didn't have so many participants here. And it's also very difficult to show that with saltiness intensity measures, but we can discuss that later. And these are the EEG results. So here it's a result on 1 electrode, but we showed that on 5 different electrodes. So this was a simple EEG recording. And we have here the 2 peaks. what we can see very quickly is that this first peak doesn't seem to change between the different solutions, while this P3 peak is appearing in some of the solutions, and it seems to not have the same time latency. So some of the peaks appear later than the other ones. So that's what we actually measured. So we have a P3 peak that appears in 3 of the solutions. It doesn't show up in the control solutions. And it appears that this peak is really delayed when we have the aroma solutions. So when we have the odor-induced taste enhancement in this solution. So what we could conclude is that there are no effect on the P1 peak, the one that is linked to the primary sensory areas, but there is an effect on this P3 peak, and especially the latency of this peak is delayed for the solution with the odor and taste together mixed in the solution. So this seems to show that the odor-induced taste enhancement is processed only in the late part of the processing, the one that is linked to cognitive brain areas and so on. So this result seems to show that this delayed latency is linked to a higher number of synapses, which would be involved in the odor-induced taste processing compared to when it's processed separately. And this was also shown by some studies, not with EEG, but just that explain that this delayed latency is linked to a higher number of synapses. And just finally, this P3 peak appear also as an interesting brain component to study odor-induced taste enhancement. So this is very specific to neuroscience, but quite interesting to have markers of some phenomenon to look at when we do evoke potential recordings. Okay, and I come back to the 2 views I presented before. So to decipher between these 2 views, the result seems to show that we are more in this classical view. We don't have effect on the P1 peak, so we don't have communication between the primary sensory areas here. We mostly have communication within these higher order areas, the ones that will induce this P3 peak. Okay, and just to finish on this concept, what we can add also in this diagram is that there is probably, because we have this delayed latency, it should be that there are more synapses. And this could be explained by a phenomenon that is called as a top-down processing, that there will be a feedback of information from the higher order areas to the low order areas, the ones that are primary sensory. And this would explain why we have this intensity increased perception. So it seems that this integrating flavor area will send information and increase the taste intensity in the primary taste areas. Okay, so now I will present you some data about more sensory studies where we look at this odor-induced taste enhancement in a normal weight and obese population. So what we know from the literature is that there is only one group that showed results on this question, and they found that the perceived sweetness of a custard dessert was increased by a better aroma, but only in the obese group that was tested. So this was interesting, and we wanted to replicate this result, but also to go further by testing also salty solutions. So we tested 43 normal weight, 37 obese, and we tried to maintain, of course, there are many in obesity, there are many comorbidities and also parameters that can bias the results. So we try to adjust as much as possible the 2 groups of participants regarding age, sex, level of study, socio-professional categories, and so on. Therefore, the groups differed mainly by their BMI, the body mass index. Yes. And we tested different aromas. So on the sweet part, we had 3 different vanillas. One of them was vanillin, then the other ones were more complex, like French vanilla, mixed aroma, and there was also ethyl vanillin, which is a simple molecule. Then we had 2 aromas, complex aromas of lychee, and 1 furaneol molecule. So this was for the sweet part, and for the salty part, we had a bacon, which was also a mixture of odorants, and garlic, which was an infusion from dry garlic. We tested that in 2 solutions, water solutions, so 1 sweet, 1 salty. And also we wanted to try this test in more complex solutions, more close to real solutions. So we tested an apple juice for the sweet part and a green pea soup for the salty part. So here we didn't use the intensity scales, just because the intensity scales, we used that in other studies before with similar aromas, and the replication of the result was very difficult. So we decided to switch to another task that is less cognitively demanding, that seems to work quite nicely for odor-induced taste enhancement, and we used a ranking task. So within the ranking task, we gave the participants 4 bottles. 3 of them contained the reference solutions, which were solutions with an increasing concentration of sucrose or salt. And the 4th solution was the solution with the lowest level of sucrose or salt and the aroma. And then they had to rank according to the taste intensity, either sweetness or saltiness. So there were 4 possibilities in the rankings that are displayed here. If the participants ranked the solution with aroma in the 1st position, then there are no odor-induced taste enhancement. If they rank it in the 2nd, 3rd, or 4th position, then there are different levels of odor-induced taste enhancement. So here are the results. These are the results in water. And we can see that basically we see many odor-induced taste enhancement in the water solutions. So this occurs for almost all the aromas, but to different levels. So the aromas that were less working were the lychee ones and also the furaneol. So here we can see that lychee is mostly working only in normal weight participants, while furaneol is only working in the obese participants. And the other interesting result is that on the salty part, we have here this result that is interesting, that participants with obesity rated in more than 30% of the participants ranked this solution at the 3rd or 4th level. So they perceived odor-induced taste enhancement that is quite high. And these are the results for the more complex solutions. We can see here that there are much less significant results, which is expected also because we know that in this solution there are other interactions. So interactions between taste and taste, or between odors and odors, that can modulate the taste intensity also. So what we see is that in the obese population, so sorry, I mixed the significancy here. So it's reversed between the 2 groups. So these groups had a much higher perception of the sweetness intensity than the normal weight group. And we can see here that the 2 groups perceive the bacon enhancement, the saltiness enhancement with the bacon. So just to resume this result, with this ranking task, we had shown several odor-induced taste enhancement. We can see that there are two results that are interesting, where we have a high perception of odor-induced taste enhancement in the obese group. So 35% rated this bacon odorant as inducing an increase to a solution until a level of a solution which had 40% more sugar. And this was the case also for the vanilla odorant, vanillin odorant in the apple juice, where the participants rated this solution as a solution with 25% more sugar. So just to conclude, so we saw that there are less aroma that produce the odor-induced taste enhancement in complex beverages compared with salty, water beverages, due to multiple taste, and odor taste interactions. And on the group comparisons, I will not go through the results again, but basically there were more odor-induced taste enhancement depending, this odor-induced taste enhancement depends on the aroma and basis. And what we can see is that the obese participant seems to outperform the normal weight on the strength of the odor-induced taste enhancement for at least two of the solutions we tested. So just to explain this result, it seemed that the experience effects is important in this association between aroma and taste. And we suppose that the obese group had more experience with some of the aromas compared to the normal weight group, which can explain this difference in the rankings of the solutions. Okay, so just to conclude, we are still working on this aspect and looking more deeply into the brain mechanisms between the populations. And I would like to thank the students who participated in these studies. And also just for the EG studies too, I would like to thank the labs who also participated in that and the Smell and Taste Clinic in Dresden.
So thank you very much, Charlotte.
Open to question, yeah.
And yes, are there some questions? I think we have a strong delay and likely everybody is extenuated. Yes, it's time for the question. Congrats.
So use of bacon in sweet solutions. Yes, we haven't tried that. There was actually one study by David Lab, who tested, I think, a beef stock solution in a sweet to see whether it could increase sweetness, and it worked. So I'm not sure how they explained that, but it seemed that there can be some association that are not expected in the European culture. For sure, some of the aromas that appears as salty in our in France, for example, are not working in other countries. So this is culturally dependent.
So an important remark.
But you mentioned bacon in sugar solution. No, I don't think so. Bacon was tested in salty water and also in green pea soup. So here, sorry, maybe I went a bit fast on these slides, but yeah, so bacon was tested in green pea soup and in salty water.
Thank you. Is there some other questions? So I think that everybody is
Okay.
extenuated or is hungry. So thank you very much, Charlotte, for this very complete presentation.
You're welcome.
And to all the lecturers, if they are still here, because the presentation were really very interesting, and it is the good reason for why we have some delays. Yes, Laetitia, thank you. You're very active. Yes, everybody will receive the slide and also the replay of the webinar.
Okay. Thank you very much.
Okay, so thank you very much, everybody. Bon appétit, dirons-nous, and have a good day. Bye-bye.